Hello. Did you know that in Finding Nemo, Nemo was actually supposed to have a second mother? Who might that be? Who might that be, you ask? Well, I'll let you know in the next slide. But for now, I'll tell you about the theme of my video today, which is the sexual plasticity in the brain and gonads of the bamboo leaf wrasse. But to answer your firing question, who is Nemo's second mother supposed to be? Well, the answer, Marlin. That's right. In real life, clownfish actually, when the mother clownfish dies, the father clownfish actually steps up and goes through a sex change process in order to keep the bloodline going and lay more eggs. So, after the Barracuda absolutely demolishes Marlin and Nemo's bloodline in the movie, um, Marlin should have stepped up, but he didn't. And that probably would have changed the plot line immensely. But yeah, a little self-introduction. My name is Luke May. I'm studying biology and Japanese through Northern Arizona University's interdisciplinary global programs. Great program, by the way. You all should be very proud. And whoever is interested, definitely take a look. Experience of a lifetime. But anyways, I studied at Kyushu University in Fukuoka, Japan. Very countryside, very rugged, not quite the image of Japan you might think, but it was a great experience. Um, and so for my internship experience, I actually got to build on the research of the lab that I joined on the species of bamboo leaf wrasse, which have the capabilities to change sex bidirectionally. So coming into this lab, they had a lot of research on the female or the male to female sex change process, but not quite the female to male, which is kind of what I based my project around. Their research had to do with the aromatase gene, which basically encodes for the enzymes that convert androgens like testosterone into estrogens, um, which explains the male to female testosterone converted to estrogen. Um, that explains a lot of that sex change process, but they really didn't know why females or yeah, females changed into males. So basically, using their previous research on the aromatase gene, they wanted me to look inside the brain, um, the aromatase gene in the brain, and see if that had a vital role in the female to male sex change. Because they had found that something called DNA methylation in the aromatase gene, um, when it actually inhibited the male to female sex change, and they wanted to see if there was DNA methylation differences between the aromatase gene in the brain and in the gonads, which is what my project was. So yeah, I did various protocols in order to get those results and measure those differences. Include some fun field work of fishing and actually dissecting a fish, which was kind of scary, but I picked it up pretty fast, I would say. So proud of that. But yeah, I just want to talk about some key takeaways of my experience. Mental resilience, I really um, went through a lot, and it was my project was very self-guided actually, which it leads into my other point of being communicative. I wasn't quite communicative with my supervisor or my lab mates about what I needed or what I wanted to do because I was scared of being a bother. But I would say don't be scared of being a bother. Language barrier is really hard to overcome no matter how long you study a language, because you know the unknown. You've never experienced some of these things ever in your life. So um, it's just It's really a mental battle and a journey of self-development, um, that you learn by yourself, kind of what you need, where you want to go, what you want to do, and how to get there. Um, so yeah, the last thing I wanted to show was my view. Basically every day while I was walking to the convenience store in Japan, which was like my third home other than the cafeteria being my second home. But yeah, this was my view, and thank you so much for listening to my video. And I hope you guys come and listen to my um, presentation at the summit. Thank you.